restriction enzymes, which are found in bacteria. They protect against viral DNA attacks and protect against their own restriction enzymes by chemical modification, which is methylation of their base pairs. They are sticky, short, single-stranded overhangs or blunt ends. Those are all possible options. And then we have DNA ligase, which joins or anneals DNA. If the source and vector have complementary sticky ends from being cut by the same restriction enzyme, DNA ligase can anneal them. If the source DNA is PCR generated, the DNA ligase can join amplified DNA to specialized vectors.
What's that word?
Recombinating.
Recombinating. Okay. So this is specialized strands of E. coli, which is also known as yeast. They have been designed for cloning using homologous, thank you, recombination. So this process allows foreign DNA to be inserted into vectors or chromosomes, and then usually through cell transformation, the recombinant vector is inserted into the host cell. So the design the insert DNA fragment to possess short homologous sequence with target DNA and active recombinase enzymes that flip while inserting DNA insert into the vector. So there are problems with this. The recombinant DNA introduced into suitable host for replication may be one. It often yields mixture of recombinant constructs in which only some cells contain the desired gene. And also, selection can identify correct recombinant colonies. For example, a change of color of colonies or antibiotic resistance.
That makes sense, because if like it wasn't recombinated correctly, like the cell would pretty much shut down and lyse. Yeah, that makes sense. And that's kind of like the next slide that talks about like cloning vectors, and there are several different types of those, known as viruses and cosmids, which are huge lambda-phage COS sites in artificial chromosomes. These cloning vectors use dependent on size of fragment to be cloned and hosted. And specifically to talk about the plasmids, they contain the ampicillin resistance gene. So that your host pretty much knows that you have received the plasmid, which is the translating gene. The plasmids contain blue, white, and color screening systems to select for recombinants. So you can kind of mismatch the way the DNA is inserted based on the tRNA and mRNA coding sites. Also, to add on to that, the plasmids contain an ORI, or point of origin of replication, so that it can break off once the ligase and the transcriptase reaches final destination site. And in order to ensure that it has a multiple cloning site, so that multiple of these plasmids can be gened, it uses the LAC-Z gene, which encodes for lactose degrading, so it pretty much degrades once the cloning is done. Yeah.
Just an overview from the figure. For cloning the plasmid into the vector PUC19, it's not super hard to do. You create sticky end products using digestion of EcoR1, which is a restriction enzyme, and use PCR to amplify your fragments. You then put these fragments into your plasmid and transform your host cell using divalent methods. After this, you grow your host that contains the vector that you want, which in this case is ampicillin resistance, and you put it on a medium. And if it grows, it has the resistance, and if it doesn't, then it does not have the resistance. Most of the time, whenever you're doing something like this, you add something else that'll help you identify whether your plasmid has taken up that gene. In this case, for this example, it's specialized galactose that produces a blue product. If there's no blue product, then the plasmid recombination was unsuccessful, and if so, then it was.
So I'm pretty sure that this one is these are the steps for PUC19. So first, you use a restriction enzyme, or restriction enzyme with cut site with MCS is chosen. Okay, so you pick the restriction enzyme that you're trying to cut out. vector and foreign DNA cut with that enzyme, and then you insert the DNA that you want into that cut site with DNA ligase. And then you use insertional inactivation with a gene called LAC-Z, which is used to detect cloned DNA to find it. Transformants plated on media containing ampicillin and X-gal to detect B-galactosidase activity. The cells that contain a vector with insert are white because there is no B-galactosidase formed. You don't study other vectors, just the PUC19 cut. And then there's YACs, yeast artificial chromosomes, which are used for cloning directly into yeast, such as Saccharomyces cerevisiae.
Y'all are so good at pronouncing these words. I didn't know I had to.
So unnecessarily long.
I know.
Yeah.
So the last slide of molecular cloning is about hosts for cloning vectors. So the most useful are non-pathogenic, and they're easy to grow and transform with engineered DNA. They're going to genetically be stable in culture, and they must replicate vector, so plasmid or like he mentioned. It's helpful if background information on a host and tools for manipulation exists. E. coli, which would be a modified strand, or Bacillus subtilis, which is protein secretion, or that one, eukaryotic genes. So yeah. And some applications require plant tissue, insect cells, or mammalian cells.
Moving on to reporter gene and gene fusions, which is our next of the three topics for the day. Reporter gene fusion is pretty much the way that coding sequence works from a reporter once it's fused with a regulatory region from another source, and this forms a hybrid gene, kind of like when you have two different animals and they mate and you have a hybrid animal. That's the general consensus of that. This regulation is studied by assaying a reporter, so it's pretty much measuring different conditions sensed by a regulator. Specifically, the reporter gene encodes protein easy to detect and assay. So once the reporter gene is inserted into the DNA fragment, it can help protein manufacturing in the cell, make it a lot easier. And this reporter gene can be fused with other genes to even help you promote other genes and other different types of expressions. This was first demonstrated by a scientist using LAC-Z from E. coli, and he encoded beta-galactosidase. And this reporter gene was used for lactose catabolism. So it pretty much cleaved it and it gave it that blue color, which is how it was first demonstrated. I can't read the rest of the slide, but it does talk about more fluorescence and stuff like that, and prokaryotic and eukaryotic cells. And they also use reporter genes as well to ensure that they have proper transcription and translation. Awesome sauce.
It's cool. It's cool.
Moving on, making products from genetically engineered microbes is kind of the next topic that we're going to be discussing. So, first off, we're going to be talking about somatotropin and other mammalian proteins. So, biotechnology is the use of living organisms for production of valuable products. Basically taking those genetically engineered microbes and taking products from them. Many mammalian proteins have high pharmaceutical value, but are very costly to purify because of low amounts in normal tissue. So microorganisms used instead. An example of this would actually be microorganisms that produce insulin. Nowadays, majority insulin for diabetes.
That's cool.
Well, this next slide is a figure,
Shut it.
which is showing the process of cloning and expression of bovine somatotropin. So really what you're going to do here is you're going to get BST mRNA from a cow, which is your bovine somatotropin mRNA. and you're going to convert it to mRNA, or you're going to convert the mRNA to cDNA using reverse transcriptase, and that turns into cDNA, and then you insert that cDNA into the plasmid of an E. coli cell. Now, once you put that back in, it can be used for commercial production because it increases the milk yield from cows.
So yeah, that pretty much concludes our lesson today. Thank you so much for joining us.
Wait, let me show this one more.
You guys see it?
And there's our
We didn't finish all the concepts in this 20 minutes, but we did get through 3 of the 5.
Okay.
yeah. Thank you for joining us. Have a great day.